All right, so we're live. So welcome everybody to the first 2017 edition of Micro Seminar, the last of the Obama presidency, sadly. But today's speaker is Vaughn Cooper. It's going to be, hopefully, I think it's going to be a really interesting talk, and it's, again, the first of a lot of, we've got 12 great seminars, I think, scheduled for this year, so just keep following us, and we've got some great people. The whole schedule, or everybody who's scheduled, is up on the WordPress website right now. So today's speaker is Vaughn Cooper. Vaughn is, the best way to describe this without kind of me going, he is the living embodiment of my imposter syndrome, because everything I do, I just look at what he's done, and it's incredible in a variety of different ways. So he started as a undergrad at Amherst College with Paul Ewald, actually. I think he'll be talking a little bit about that. And then went and did grad school at Michigan State with Rich Lenski. Vaughn was actually the first. There's an epistatic interaction in the Lenski lab between the last name of Cooper and Lenski. I mean, Lenski helps no matter what, but there's a bunch of different Coopers from the Lenski lab. He was the first one, I believe, and did some great work there. So after Rich's lab, Vaughn had a little bit of a wandering in the desert of a time. So he was a Michigan Fellow, which is a kind of a hybrid postdoc PI position, where he was, that's where he, in his later time there, became interested in biofilms in the La Puma lab, and kind of started the work that he's going to talk about today, I think. So after being a Michigan Fellow in 2004, Vaughn started his own lab at the University of New Hampshire, which went until 2015. And in 2015, he moved to the University of Pittsburgh. Has not gotten his face plastered at the Newark airport yet, but I'm sure it's only a matter of time until that happens. And lastly, the reason if that's not kind of impressive enough, and if everything's not impressive enough with what he's going to talk about today, Vaughn's run the Ironman like multiple times and finishes, and it's crazy triathlete. So it's amazing what he's done and what he's going to do, and I think it's going to be a really great talk today. So with that, I just want to mention anybody in the Hangout, please mute. You guys are all good right now, but anybody that comes into the Hangout, please mute. And with that, I will say goodbye for 45 minutes and give you to Vaughn.
All right. Thanks, Dave. That was too generous. I'm going to screen share here, and you can see my desktop, I hope, and here we go. So you probably saw the title slide on Twitter, but if not, I'm VS Cooper on Twitter, and a lot of the material that I'm talking about is either at some links on the website, which is on top, and I want to start by acknowledging support from the funding agencies on the bottom. I also really want to thank everyone here tuning in, and especially the organizers of Micro Seminar, Jen Biddle, Cameron Thrash, Dave Baltris, and all of you for attending. It's really a huge honor. This is a great opportunity to talk to you all. And I think it's a great opportunity to be doing this sort of work. Technology has allowed us to share our science without traveling, in a fairly democratic way. And it's a wonderful time, maybe the best ever time, to be an evolutionary biologist or a microbiologist, which is really the theme of today's talk. So I'll talk briefly about evolution experiments in general and why they're powerful and how I got interested in them, how we need to understand evolution in biofilms, and we'll focus on Burkholderia and Pseudomonas biofilms in particular, motivated by their role, their terrible role in chronic infections of the cystic fibrosis airway. And then finally, if I have a moment, I'll talk about how I think we all need to consider how to develop hands-on experiments to teach science in a hands-on way. And we've been working to do that, to produce a curriculum using evolution in action to teach the concepts of evolution and heredity. So I started out as a fisherman, still am, and really wanted to do stream ecology. So when I wound up at Amherst College, I was doing some field work in a lab, and I thought, right, I'm going to do predator-prey dynamics. And then I started learning a bit more in an evolution class about these explosive adaptive radiations of cichlid fishes in the Rift Valley lakes in Africa, where the lakes were formed and some lucky founder species exploded into a variety of huge number of species. So essentially nearly 2,000 species evolved in a relatively short period of time in the apparent absence of geographic barriers. And I said, right, so not just ecology, I want to study evolution. And so I thought, well, maybe we can study some dynamics that indicate selective pressures. And my advisor, Paul Leewald at Amherst College, said, yeah, Vaughn, that's great, but you're never going to get that done as an undergraduate. And you're never going to get that done during a PhD thesis either. So why don't we try and do some experiments that are a bit more feasible? And so he had started this project by a chain of using a series of undergrads involving the gypsy moth, which had been really devastating New England forests, and a virus, a naturally occurring virus that had actually been kind of developed as a pesticide called the nuclear polyhedrosis virus. This is a baculovirus that literally liquefies the gypsy moth caterpillars. And their question was, if you could vary the transmission of this virus, might we observe changes in the evolution of virulence? And that concept of that virulence evolves is really what Ewald is known for, I think. But this experiment that I got sort of seduced to do turned out to be incredibly powerful, not just for understanding virulence, sort of the virulence of the baculovirus, but also some principles about how pathogens evolve in general. And so what our experiment involved treatments of viruses transmitted early during the infection and also late during infection. And the early transmitted viruses tended to favor rapid replicators, which became more lethal. And so on the left, you can see that the early transmitted group were killed more larvae during a bioassay than the late transmitted virus. And the stock virus, which is genetically heterogeneous, called GIP-check, was intermediate. And so we learned that selecting on the early phases of transmission might increase virulence. But what we also learned, I think, is maybe even more interesting, and that was that the early transmitted viruses tended to kill smaller insects. And so their net productivity per infection was lower. And so in this experiment, we saw that virulence could evolve, and it could also generate some life history trade-offs, both for the virus and for the host. Okay, so I was hooked now that we could actually study evolution during experiments. And that eventually brought me to Rich Lenski's lab, who's probably still maybe the best place to do this. My PhD career in Rich's lab was from the long-term evolution experiment generations, 14,000 to 22,000 generations. It was a tremendous time. I learned a lot. I worked with some tremendous people, including another Cooper, Tim Cooper, a number of other great postdocs like Santi Elena and Greg Veliser and Brennan Bohannon and so on. And we were fortunate to help discover some of the first mutations that drove adaptation in the Lenski experiment by really by a combination of luck and a lot of sweat. This is pre-sequencing. But I really, from my undergrad, had been really committed to studying pathogen evolution. So I made my way to Michigan and realized that actually most microbes don't live in well-mixed cultures. Not in nature, and really the background of our field had been studying bacterial growth that was probably non-realistic because most microbes live on surfaces, as you know. And so when I finally made my way to John LaPuma's lab at Michigan, it was pretty clear that these biofilm-forming organisms that were colonizing the lungs of cystic fibrosis patients that John and others were taking care of grew a lot differently. They were highly tolerant to antibiotics. They produced a lot of biofilm, and that biofilm they formed biofilm on a variety of surfaces, including the CF airway. And the outcomes of these biofilm-associated infections were not good. So today, biofilm-associated infections are known to increase mortality and are incredibly costly, resulting in easily over 500,000 excess deaths per year and many billions of dollars of excess investments in healthcare costs. Okay, so this poses the challenge. How do we study then evolutionary dynamics in biofilms? So what are biofilms? In fact, biofilms are not just a single phase of growth. They represent a fairly complex life cycle whereby bacteria attach to a surface and start to elaborate a growth pattern where they secrete polysaccharides and extracellular DNA and proteins that bind them, produce slime. These biofilms mature, and then they become more resistant to antibiotics and host immune defenses. And as those biofilms mature, they also tend to encapsulate phenotypic diversity and perhaps also genetic diversity. That is, the bacteria evolve during the formation of these biofilms. But I guess I'd argue that a biofilm that doesn't disperse is a dead end, and so that means there must be at some stage a dispersal phase where cells leave the biofilm and revert to a planktonic stage to go colonize and repeat the cycle. One process that's known to drive some of these transitions is quorum sensing, which is this density-dependent process mediated by small molecules that bacteria secrete and regulate diverse gene expression. So in a planktonic phase, the cells are producing these quorum sensing molecules, and they are relatively dilute. But as the bacteria attach to the surface, their relative cell density increases, and so the perception of those quorum sensing molecules increase, and that activates biofilm-related traits. And I want to acknowledge Megan Kudrowski, a postdoc in Jim Baumberger's lab, for this great animation. So we want to study evolution in biofilms. How do we do this? There have been a number of attempts here, but the problem is that most models of biofilms in the lab can't be reconstituted or periodically sampled and archived. That is, you can grow a biofilm, but then you must destructively sample it, and it's impossible to rebuild the state that the bacteria evolved in. If we want to study biofilms in hosts, that's often a challenge because the biofilms might be in the lungs, and if they're sparsely sampled, it's tough to represent that complete community. Yet another big problem is understanding the demography of biofilm population assembly. We've not made much progress here, but this is an active focus of our lab. And then finally, we really don't understand the process of dispersal and recolonization. So these were goals to lead us to develop a new model, a ridiculously simple model, where we simply put a bead in a test tube and asked bacteria to stick to the plastic bead. So at the left here, you can see there's a plastic bead, 7 millimeters, in a test tube, and we inoculate a tube with a clone. And after a day, the bacteria colonize the bead, and then that bead becomes the population. That colonized bead becomes the population to add to a new bead. And the cells must leave the old bead and colonize the new bead, and then the new bead gets transferred every day. And so this sets up this biofilm life cycle where cells attach to a plastic bead, form a biofilm, the bead gets moved to a new tube with new fresh media, then they have to disperse and colonize a new bead. So the work that I'll be telling you about using this system to try and understand some basic dynamics of adaptation in biofilm is focused on this organism, Burkholderia cenocepacia. It's a gram-negative beta proteobacterium. It's a member of the Burkholderia cepacia complex. Burkholderia cepacia complex bacteria cause a relatively low number of infections in persons with cystic fibrosis, but they're associated with significantly worse outcomes. They often, patients who are infected with Burkholderia cepacia complex bacteria are often removed, from lung transplant candidate lists. So it's a major concern for the CF community. And then towards the end, I'll talk briefly about some work that we've been doing for a while with Pseudomonas aeruginosa, which is a far more dominant pathogen in the CF community. So we started some pilot experiments with these organisms in our test tube model, and we found that very quickly diversity evolved. And so here's a snapshot of some of the morphological diversity that might arise in biofilms. And we'd expected this, right? When bacteria grow in biofilms, the surface itself segregates different lineages. And so you can imagine that simply spatial structure alone preserves genetic diversity. But it was very surprising to us how common and how rapid this diversification happened. So Stefan Poltak, early PhD, really one of my first PhD student, actually my first PhD student at University of New Hampshire, started a 6-month evolution experiment with Burkholderia senescipacea. He established 6 replicate biofilm populations and 6 planktonic populations. You'll see that adds up to 12, which is the Lenski number. And studied this evolution of morphological and ecological diversity over time. And what he found is that all 6 replicate populations evolved into 3 distinct ecotypes, which he named studded, ruffled, and wrinkly. And the smooth or studded type was the majority type in all replicate populations, about 70% of the population. And these other 2 types, which you can see produce more biofilm and more biofilm still, can be seen as sort of ecological specialists that produce far more biofilm at the cost of planktonic growth. The studded type is a bit more of a generalist because it retains the ability to grow well in the planktonic phase. And that wrinkly type is found in all populations and was associated with the most biofilm in each population. So it was surprising then that evolution in a spatially structured environment would lead to fairly reproducible patterns of diversification. We wanted to understand what forces drove the emergence of this diversity and led to their long-term maintenance. And so what Stefan did was label the different types, a clone representing the studded type, the ruffled type, and the wrinkly type with different fluorophores. And then he allowed them to grow together and image them by confocal microscopy. And what you see here is a Z-stack where we're only looking at one color at the outset, the blue color representing the studded type, and this shows you that the bacteria is growing really on the exterior of the biofilm, on the top. So this implies that these sort of generalist studded types are balancing, sticking, and swimming, and in fact tend to be more late attaching. The ruffled type, when we turn on the green channel, is visible as a surface-associated type, but that is a plastic-associated type, and it's sort of filling in the gaps where the studded type is not. And when we turn on the red channel for the wrinkly, we see not so much red here, but purple. And that's because that relatively rare wrinkly type is sticking to the plastic surface and the studded type is sticking on top of it. So underneath this layer is wrinkly. And that generates some ecological synergy between the 2 of them. So one other force that might maintain diversity is some metabolic interaction. And so Stefan asked whether they might be cross-feeding one another. Oh, and just to illustrate over here, this is now the model that we think of how these types interact. So the studded type is growing on the exterior in blue, the wrinkly and the green ruffled type are attaching to the plastic surface. So we have this spatial segregation that's maintaining diversity. Alluding to the potential metabolic interactions, he asked whether they could grow in each other's supernatant. And what he found is that the studded type was secreting a good amount of resources, probably small organic acids, back into the media that it itself could use, but it could also feed the 2 ecological specialist, sort of biofilm specialists, wrinkly and ruffled. And these types were benefiting from that cross-feeding interaction from the type that was sticking on top of it. So just over the course of a few hundred generations, we saw a population that became diverse and stayed diverse by both spatial segregation and metabolic interactions. We wanted to know then what the nature of these interactions were. So despite the fact that you could see that they might be cross-feeding, you could imagine that these types are really more competitors than mutualists. And so Crystal Ellis took up some of this work with Stefan and asked whether these populations were dominated more by competitive forces or by complementary forces. So at the left here, you see isolates from the early phases of one of the replicate populations grown as clones by themselves. So these are monocultures, studded, ruffled, and wrinkly clones. And from that output, we would predict, based on their starting frequencies or frequencies added to the tube, that their output would give sort of an intermediate level of productivity. And when you actually put them together, the early population produced somewhat less biomass. This is colony forming units per milliliter bound to the plastic bead than what we'd have predicted from their output in monoculture. But you'll note that that changed over the course of the experiment, such that the late isolates actually are producing more CFUs bound to the bead than we would have predicted from their output in monoculture. So they're becoming a synergistic population where, in fact, all mutants are benefiting per capita from growing together. We also wondered whether the origin and maintenance of these types throughout the long-term 6-month experiment might involve inhabiting common niches, that is, discrete niches. And so we asked whether the wrinkly mutants from different populations might be filling the relatively same role. That is, that 5% member of the community that tends to stick to the plastic surface first. If we swap those out and put one from one replicate population into another, might we see the same output? And what we found is that, in fact, despite the fact that the wrinklies, when grown by themselves, had different phenotypes, different levels of fitness in competing versus the ancestor, when we put them in the mixed population, they formed the same role. So different wrinkly mutants are filling the same niche equivalently. And so that led us to conclude that in this long-term evolution experiment, the spatial structure generates early ecological diversity. That diversity is competitive for nutrients in space. And over time, they co-evolve to undergo a process of character displacement, where the studded type tends to now stick to the stickers, and biofilm specialists exclude and become great at attaching to the plastic surface. And in fact, some of the wrinkly types seem to be filling a particular niche, such that multiple wrinklies evolved independently can fill the same niche. So we'd always wanted to know what these evolutionary dynamics might be. So after focusing a lot on really the evolutionary ecology of the biofilms, we decided to tackle the problem of what mutational and population dynamics might be explaining this adaptation. And so we knew from theory and from what we saw in the Lenski lines, which were well mixed, at least in the early, well, are well mixed, and their dynamics over the early phase of the experiment, that you have series of selective sweeps by a dominant dominant haplotype, so that mutant A arises and sweeps the population, and then a mutant B arises on that genetic background, and that invades and sweeps the population. And then, say, another mutant arrives on that AB background, and mutant ABC, that haplotype, sweeps the population. So these are global selective sweeps. We knew from our sort of basic microbiology that our biofilm population was unlikely to follow that model. That is because the ecotypes, these different colony morphologies, arose early and persisted. And so that led us to hypothesize that we might have niche-specific selective sweeps. And so on the right here, you see this is real data now. This is our Muller diagram from the population genomics of that one replicate population we've been focusing on. And you can see actually over here on the right, a series of changes in colors in green representing mutations sweeping through the ruffled niche. And we also see in the middle here that dominant studded slice of the population, mutations arising and sweeping through the population. You have a cluster of mutations arising at once that sweeps through the population. So it seemed plausible before we started to understand the structure of the haplotypes that this model of niche-specific sweeps might have been true. But what we found when we started to reconstruct the haplotypes by clone sequencing was that, in fact, mutants arising in the studded type, so these are studded colony morphologies with certain genotypes, were then picking up mutations that allowed them to invade the other niches. So they were picking up new determining or sort of ecologically determining mutations that allowed them to invade. And over here in the ruffled types, they displace that entire earlier founding population of ruffled. And that was also happening in the wrinkly niche as well. New wrinklies were evolving from the studded type. So this was pretty surprising. This generalist population continued to spawn new ecological specialists that invaded and in some cases displaced the prior specialist. And so, of course, we had a reference genome, so we could now look to see what were these mutations. And so, although this slide is fairly complicated, I've highlighted two clusters, and these two clusters are mutations occurring in the same loci, the blue loci and the red loci, two discrete loci explaining a good fraction of the number of mutations that I'm showing you in this Muller plot. And so if we look across the population, the entire experiment of the 6 replicate biofilm populations, and plot the number of mutations per locus and their frequency in the entire experiment. So this is just a histogram of the number of mutations from the long-term experiment, frequency and versus numbers of mutations per locus. We found that, yeah, about half the mutations occurred in one and only one gene throughout the experiment. But a surprising number occurred in the same gene over and over again. And there were 2 gene clusters that were real outliers. This WISP cluster in red, which turns out to be the determining factor that produces these wrinkly colony variants in our system. And this gene that we didn't know anything about called YCIR. So for E. coli people, this is a Y gene. It's now got a bunch of different names, also called, YCR is also now called RPFR or GMR. It has another name now too, I believe, which was pretty surprising. So I just want to say that now one of the reasons why it's so exciting to do this sort of work is we can use evolution experiments as a forward genetic screen to understand the earliest processes of adaptation and also really how our organisms work. And so we've really dug into this gene YCIR and also this WISP cluster, which I won't go into more detail today. This YCR gene is super interesting. So YCIR is a gene that has a diguanylate cyclase domain shown here in the blue phase with the GGDEF domain. It has a phosphodiesterase domain denoted by this EAL domain. It's got a sensor domain. And then another domain at the N-terminus that we think is partnering with this sensor domain to help modulate signaling. And what this signaling molecule or fraction of this enzyme appears to be doing is responding to the concentrations of a quorum sensing molecule, a fatty acid, cis-2-dodecanoic acid, otherwise known as BDSF or Burkholderia diffusible signal factor, which the organism is producing and then at high density sensing by binding to the sensor domain. And so here, I've got an illustration here how we think that mutations in RPFR or YCIR are generating diversity in our system. So at the outset, the YCR enzyme has endogenous diguanylate cyclase activity, which is producing cyclic diGMP. Cyclic diGMP is the central kind of regulator of the stick or swim cell-fate decision. So when cyclic diGMP levels are low, you'd expect a more planktonic phase of growth, and when they're high, you'd expect biofilm phase. So given that YCIR has this endogenous diguanylate cyclase activity, it helps Burkholderia to form biofilms when they're attached. And then we transfer that bead with a high dense biofilm where quorum sensing is high. And that quorum sensing molecule now comes in and binds the sensor domain, and that activates the EAL domain of our enzyme, which now degrades that phosphodiesterase activity, degrades cyclic diGMP, promoting dispersal and a new life cycle phase. And so this enzyme's particularly interesting because it's sort of the one enzyme that picked up, or one gene that picked up lots of mutations in our experiment, despite the fact there are lots of other diguanylate cyclase and phosphodiesterase genes in the Burkholderia genome. And it also is this kind of seesaw where the sensor domain modulates either the stick or swim sulfate decision all in one enzyme. And we've since gotten into the details by making single mutants in this gene in different domains. And you can see here from this slide that a single mutation in the sensor domain produces the ruffled colony morphology that arose early in the experiment. Another mutation occurs in the zygonolyte cyclase domain, produces the studded variant that evolved early in the experiment. And then this separate cluster that I'm not talking much about in the wisp cluster produces the wrinkly colony morphology. And these are the colony morphologies, and these are spot biofilms. So very little differences in our genome produce conspicuously different ecological strategies affecting biofilm. And so to conclude, we find in the Burkholderia experiment that adaptive diversification is driven by mutations in relatively few loci, leading 3 coexisting strategies whose combined actions are synergistic. And so this pattern of diversification was pretty predictable. And we've repeated this experiment many times, and we always see these types cropping up, which leads us to believe that at least in our very simple experiment, we might be able to start predicting the dynamics of evolution in our model biofilms. But what was really stunning to us, something that we really didn't anticipate at all, was the extent to which the mutated genes in our lab experiment, simple test tubes, minimal galactose media, occurred in genes that also were being detected as being mutated during chronic infections of the cystic fibrosis airway. And these included genes affecting cyclic diGMP metabolism, a number of genes affecting LPS and EPS biosynthesis, genes affecting the RNA polymerase subunits, and iron metabolism. So that really prompted us to now dig in greater detail into the dynamics of adaptation at higher resolution in Burkholderia infections. So a bunch of work had been done in Pseudomonas, and it's still ongoing. We're part of some of that. But Burkholderia infections were relatively understudied until really the Burkholderia dolosa epidemic, another Burkholderia cepacia complex organism, affected the patients at the cystic fibrosis clinic in Boston. So we had some ideas that diversification and where it was occurring maybe in Burkholderia infections of CF infections, and that some of these pathways might be important. And I was really fortunate to discover a study of another Burkholderia cepacia complex species that was ongoing involving Burkholderia multivorans, which causes about half of the Burkholderia cepacia complex infections these days in the CF community. And this is a study of one patient visiting the University of British Columbia CF clinic in Vancouver over the last 20 years. She was infected back at least in 1993, and samples have been taken from that chronic infection periodically over time. And the reason why Leonilda Moreira from an institute in Lisbon was interested in this particular patient and a few others was this transition between mucoid and non-mucoid variants over time. So the black isolates here were the non-mucoid variants, that is producing less biofilm, and the red types were the mucoid variants producing more biofilm on certain plates. In Burkholderia infections, this transition to non-mucoid is associated with worse outcomes. And so Leonilda and her really great postdoc, Inez Silva, had been doing work using genomics to try and understand how this evolutionary process happened. I was fortunate to get involved because we were at the same meeting, and then I was able to get my friend Chris Waters involved from Michigan State to help us ask whether variation in cyclic diGMP metabolism might be underlying some of this variation. And so Inez sequenced all of these genomes, and together we put together a phylogeny of these isolates. So first, up here in the first panel, we can see that over time that population seems to be accumulating SNPs at a per genome level of basically 2 SNPs per year, very steadily over the 20-year history of this infection. There is one outlier here that's excluded from this plot, and that's a mutator lineage that you see over here in the phylogeny over in panel B. But you'll note that despite this steady accumulation at a population level of SNPs per genome, that I've labeled these isolates by different colors that correspond to different clades in the family tree of this species, this population. So very early on, this population diversified into separate clades, and there was a period where there was really a rapid splitting into different clades that coexisted over time. So panel C shows you really that at any given time there are multiple clades persisting in this population. And you'll note that this is really a wonderful study, but it's still really sparsely studied. We really have the power because of the length of time this infection was sampled. But this is just basically one clone picked every year or every other year, with a couple of times where 2 clones were picked because the colony morphologies look different. And when that happened, you see that those 2 clones fall into different clades in the infection. So in this long-term infection, diversity arose and persisted over time. So we can now look at what those mutations are. Most are non-synonymous, many of them that were not in coding regions of influence promoters. So this is clearly a selection-driven process of evolution. Many of the affected genes suggested pathways to adaptation to biofilms. And there was one gene in particular that caught my interest and really got me involved. And the mutated gene right here in the blue star is YCIR, or RPFR, which I was telling you earlier. And those mutations did what we expected. They disrupted the dominant phosphodiesterase activity of YCIR and increased the cyclic di-GMP levels of these affected clones that picked up that mutation. And the clones that have that YCR mutation become better adherent to CF lung epithelial cells, just as we'd expect. So what we saw in our plastic bead model suggests that mutations in YCIR can lead to a biofilm lifestyle, and that appears to have happened during this chronic CF infection as well. That's not the only trait. There were lots of other traits that were under selection, and one of the other ones that was conspicuous were multiple mutations affecting production of the LPS. And so this is a lovely LPS gel from Leonilda's lab showing mutations from the early phase leading to the loss of O antigen repeats. This has typically been thought to be a selective response that enables immune invasion. That may be true, but I have to emphasize that we also see mutations in these very same genes evolving in our lab experiments. And so this is just an excerpt from our paper in 2013, which reads perhaps the most compelling example of convergent evolution in this system and in the mutations identified in CF lung isolates are the many mutations in genes evolving LPS. So this suggests that adaptation within biofilms may lead to the loss of the O antigen repeats of the LPS simply because of interactions between the bacteria themselves and sticking to the surface, and not so much in response to the immune system. So another feature that was pretty obvious from this one patient were the fact that multiple genes were hit more than once. So this finding that you get repeated non-synonymous mutations in the same genes provide really strong evidence of selection. And I want to focus on just one of these right now. It's a 2-component regulatory system called FixL. That FixL gene was identified as one of the heavy hitters in this Burkholderia dolosa epidemic led by Tammy Lieberman and Roy Kashoni's lab in collaboration with Greg Preby at Boston Children's Hospital. They've been really focusing on this gene lately, and just have a very recent PLOS pathogen paper on this, showing that this enzyme is under selection, perhaps because of adaptation to growth in anaerobic conditions. And so since over the last couple years, our interest in studying evolution during Burkholderia chronic infections has gotten out, and we've been interested in collaborating with a variety of different folks who have longitudinal series of isolates. And what we found just in the last few months are fixed cell mutations in different species, in different geographic regions, all apparently leading to common phenotypes. So we have mutations from another epidemic in Portugal, another series of infections in Portugal, a cluster of infections in Michigan, the one in British Columbia that I mentioned previously, and of course the first study of the Delosa epidemic in Boston. So it leads us to ask, why is there such strong selection for mutations in this FixLJ operon? We think it's an iron sulfur cluster binding regulatory element that's analogous to ANR in Pseudomonas, and we think that that's enabling growth at low oxygen conditions and enabling persistence when buried within biofilms. So we hypothesize that the fixed mutants might be living at the core of these airway biofilms. And so in some sense, these fixed mutants we don't think fix in the population, but rather contribute to heterogeneity with infections. And we think that that heterogeneity is important. In fact, Greg Prevey has shown that the presence of a fixed mutant was associated with worse prognoses in their epidemic. And we've also asked that question in our experiment, well, not experiment, in the retrospective study as well. And what we found is that that period of diversification linked to mutations in YCIR and FIX and other loci was associated with a period of greater reduction in lung function in this one patient. So as the Burkholderia multivorans population expanded, the microbiome diversity within that CF infection declined, and that was associated with worse outcomes. Now, although these are plots of airway function over time, although the data are well fit by a linear model, this logistic function with a steeper drop in the middle is a statistically better fit. So this is an important question, I think, for the field. How does diversification, not just growth in number, but genetic diversification of the dominant infection associate with lung function in CF? So I know I'm running a little short on time, so I'm going to talk very quickly about some work we've done with Pseudomonas aeruginosa. I don't want to leave it out because it's such a big problem in the CF community. We conducted exactly the same experiment with Pseudomonas aeruginosa strain PA14, and we observed a zoo. We've described our efforts to understand the zoo in a recent J-Bacteriology paper. But we did find that this zoo of diversity is associated, again, with variation in cyclic diGMP levels, that the mutants differed in the way they attach to the surface, that some of those interactions become synergistic, that mutators evolved in all populations, and that contributed to all that diversity, and we did not see mutator populations in planktonic controls. And we've also been able to identify some of the mutations that are drivers. I'm going to skip what mutations were drivers, except to say that we think also it involves variation in oxygen sensing in a gene that's much like FIX. So we have multiple mutations occurring in the same populations. So you have mutations that are associated with one phase and mutations that are associated with another phase, where some mutations improve planktonic growth and other mutations abolish growth in anaerobic screw cap vials. So these are just it's a tube with no oxygen, very little oxygen. And you can see some of these mutations abolish the ability to grow in that low oxygen conditions, and others actually increase it. The last piece that I want to emphasize with the Pseudomonas is that we think that even though these mutator populations have a great deal of diversity, a lot of that variation occurred in relatively few loci. And so we're optimistic that studies like this are going to help us winnow down the zoo of mutations that we see even in mutator populations to help predict evolution. I've been rushing through that Pseudomonas section because I want to end on the highest note, and that is we can use evolution experiments to teach evolution and teach microbiology and teach heredity. So for the last several years, we've been working on a program which we now call Evolving STEM. It's been driven by a great former a master's student and coordinator in my lab, Taylor Warren, and two just tremendous teachers at Winnicott High School in Hampton, New Hampshire, Mike Handwork and Shani Scarponi, who agreed to test out this curriculum instead of a standard evolution curriculum. And what we did actually at the outset was split the students. Half the population, half of the classes didn't get our curriculum and half did. And we measured learning. And then we allowed those sort of deprived students to get our curriculum the second time, and we merged this in a second week, and we measured learning again. And we used not Burkholderia or Pseudomonas aeruginosa, but rather Pseudomonas fluorescens, which is perfectly safe, a terrific biofilm former, and it also undergoes this process of adaptive diversification within a week, which is great for a classroom experiment. And the results of our study were pretty compelling. The different groups had no difference at the outset, but those that got our curriculum learned, did better on our assessment than those who didn't. And then when those students got our curriculum, they caught up. So the students learn evolution concepts better by doing experiments. And importantly, they also, in a survey, appear to be more motivated to consider careers in STEM education. So we're really keen to keep this curriculum going. We're reviving it here in the Pittsburgh area, and I'm happy to say that it's still ongoing back in New Hampshire. So just some big conclusions. We tend to see, despite the diversification in these models, relatively few genes driving a lot of the diversification. And we think that's because large populations are empowering natural selection to pick the very best mutants, often in the same pathway or gene. We're finding that mutations are occurring in common between laboratory models and in infections, which suggests that bacteria are adapting by interactions with other bacteria, by mechanisms that improve adhesion, improve their ability to tolerate anaerobic growth, and regulate that important stick and swim life cycle decision that includes dispersal. We think that work like this is going to help us start to predict phases of these infections, and hopefully shed light on new strategies for treatment that allow us to steer the infection or the microbiome in general, or even the immune system to help manage these infections. And finally, what a great time is it to be a microbiologist and evolutionary biologist, where we can use genomics and technology that allow us to study bacteria really as organisms rather than as entire populations to learn more about how they affect our lives. Thank you very, very much.
All right, that was awesome. I like the diversity of topics too, and bringing in the education there it was a great example of what we can do, and I think a lot of us are trying to do as well. So any advice that you have for us teaching those types of experiments to undergrads, I think we would all welcome as well. I've got a couple questions, but I'm gonna scout everything for, if anybody has questions out there on YouTube, feel free to tweet them in, feel free to write them on the YouTube chat. I'm trying to figure out how to monitor that with a small computer screen here, but I'll take a look and see if the questions are there. If you're in the Hangout, feel free to unmute and ask questions as well. I don't see any direct tweets at it, so I'm gonna just ask a question to begin with. So I'm interested in the ruffled versus wrinkly versus everything, because pseudomonads are you just isolate them from nature. They're all sorts of these morphotypes. So given what you've seen just in these small lab passage experiments, what do you think? Because I see ruffled spreader guys out just from nature. There's some definitive pseudomonads that do this as a diagnostic type. What do you think they're doing out there in nature when they do that?
I mean, I think that I think they're good at sticking. I think that you know, if we can survey the actual population of natural pseudomonads or Burkholderia or other sort of plant-associated bugs, we're going to find a variety of strategies driven by relatively little genetic diversity. I mean, of course, they're going to be diverse, but what this is showing is that a mutation in a key regulator can go a long way. We're also finding that if we take those types and evolve them, these sort of ruffled types, and evolve them, they can regenerate that diversity, albeit by sometimes different pathways. I think that these guys that that show that morphology are effectively locked in surface sensing. So that's what you've isolated, is a high biofilm former that for now is genetically or at least phenotypically locked in its biofilm state. Sorry, not hearing you, Dave.
I'm still muted. All right.
Yeah.
So the other question is related, but a separate topic that I think I've mentioned in the past. Well, I'll question, and then there's a Twitter question online.
Okay.
So just quickly, in terms of, I think it's a unique thing that you can do maybe. So we have a handle that there's different cyclic diGMP pools within cells potentially, given that there's all these sensors and they seem to do different things. I think you might have a unique way, since you have these mutants, the ancestors and the mutants, to actually maybe spatially measure pools within the cells, or see something like that. So have you thought about that, or can you speak to that at all?
Yeah, it's a terrific question. It's not an easy question to answer, but what we're trying to do now, so what we've been doing is taking these populations apart, measuring cyclic diGMP levels of the mutants with these phenotypes. And I didn't get into the details, but the high biofilm forming colony morphologies do have higher cyclic diGMP levels, but that's from growing them by themselves. And the challenge is to grow them together and then sort them quickly, grow them in situ, sort them, and measure pools and also expression patterns from those sort of immediately sorted cells to preserve that nascent structure. The other thing we're trying to do is deploy some biosensors that have been developed for cyclic diGMP levels to see how that variation affects the coordination of the community. But I want to point out that this is a tricky question, because we find that those levels, so we often find that we find high levels of cyclic diGMP actually in the initial planktonic phases, and then once they're bound, those levels start to drop. And so sometimes you get these counterintuitive patterns with high levels at the sort of first few hours of growth, which you would associate with the attachment phase. But that's, of course, driving that attachment to start.
All right, some Twitter questions. So Akos Kovacs, I think there's the group there watching over in Europe. So their question is, how do you define synergy between evolved ecotypes?
Yeah, it's a great question. So this is a really nice application of the price equation that Leroux and Hector published something like 01 or 02, just showing that it's pretty easy then to grow different mutants by themselves and measure their properties, and then track those mutants or different types' behavior in mixture, and ask whether per capita they are more productive growing by themselves or growing together. And so it's actually relatively simple math, and what we find is that even though, say, the Wrinkleys produce a lot of colony forming units by themselves, per capita they're doing better in mixture, accounting for the number of cells that go in at the outset. So you do need to know how many you have at the start and exactly how many you have at the end. Cool.
Another question from Twitter, Tara Levine. So she says it's interesting about the loss of LPSO antigen repeats in biofilms. Can you say anything more about that and why it's happening?
Yeah, so we think that actually it's improving attachment. That this is allowing better cell-to-cell contact. So as one type attaches to another site, we suspect that those repeats might actually be interfering with the attachment patterns, might also be interfering with the process of secretion of the polymers that allow them to build the biofilm. It might actually simply be a metabolic trade-off. This is a really, so I've given you three hypotheses. I don't know which one, but this seems to be a pretty recurrent phenomenon, both in vitro and in vivo, and it's an important thing to sort out.
All right, anybody else on Twitter, Hangouts, YouTube? All right, with that, I think just want to thank you once again. That was great, and again, Thank you again, Vaughn. I wish everybody could clap, but
no, Dave, I want to thank you and everybody else who worked so hard to keep this going. Hopefully we all can keep spreading the word, and thanks again for the very kind invitation and introduction.
All right.